So hey friends, we are going to revise our lesson molecular basis of inheritance, and in this would be highlighting the main points of this chapter. Okay, I will annote. So in the previous chapter, you have learned the inheritance pattern and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material at least for the majority of organisms. In class 11, you have learned the nucleic bases are polymers of nucleotides. At least for majority of organisms, nucleic acids are polymers of nucleotides. Deoxyribonucleic acid DNA and ribonucleic acid RNA are two types of nucleic acid found in living systems. DNA acts as genetic material in most of the organisms. RNA, though it also acts as genetic material in some viruses, mostly functions as messenger. RNA has additional role as well. It functions as adapter, structural, and some cases as catalytic molecule. In class 11, you have already learned the structure of nucleotides and the way these monomers units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, and the process of making RNA from DNA, that is called transcription, and the genetic code that determines the sequence of amino acids in proteins, the process of protein synthesis, translation, and elementary basis of their regulation. Of complete nucleotide sequence of human genome during last decade had set in new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent sections, we will understand that why it is the most abundant genetic material and what its relationship is with RNA. The DNA. DNA is the long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotides referred to as base pairs present in it. This also is the characteristics of an organism. For example, a bacteriophage known as Phi 174 has 5386 nucleotide. Bacteriophage lambda has 48,502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pairs and has haploid content of human DNA is 3.3 into 10 to the power 9 base pair. Let us discuss the structure of such long polymer. Structure of polynucleotide chain. Let us recapitulate the chemical structure of polynucleotide chain, DNA or RNA. A nucleotide has three components. A nitrogenous base, pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group. There are two types of nitrogenous bases: purines, adenine and guanine, and pyrimidines, cytosine, uracil, and thymine. You can learn it by cut for pyrimidines. Okay. Cytosine is the common for both DNA and RNA, and thymine is present in DNA. Cytosine is common in both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA. At the place of thymine, a nitrogenous base is linked to the OH of first carbon pentose sugar through N-glycosidic linkage to form a nucleoside. Here you can see a nitrogenous base is linked to OH of first prime pentose sugar through N-glycosidic linkage to form a nucleoside. When a sugar plus base is present, then it is called nucleoside, okay, such as adenosine or deoxyadenosine, okay. Guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is linked to OH of 5' carbon of a nucleoside, 5' carbon phosphate group is linked nucleoside through phosphodiester linkage, a corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Two nucleotides are linked through 3 to 5 phosphodiester linkage to form a dinucleotide. Two nucleotides are linked through a 3' to 5' phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free, here you can see like at 5' phosphate group is present, and when I would number, then this is 1, 2, 3, 4, and this carbon is 5, okay. So here you can see like at 3' what is happening, 3, and this is 5. So you can see like phosphate bond is formed between 3 to 5, okay, direction. And this one is the sugar, this one is the base, and this is the phosphate, the 3 components which you require to make a nucleotide. And the direction, here you can see this is 5 phosphate and this is 3 hydroxy, because when I will number 1, 2, 3, 4 and 5, here you can see 3 is free and here 1, 2, 3, 4, 5. So here 5 is free. So this is 5 and this is 3 hydroxyl group. Okay. Phosphate moiety at 5th end of sugar, which is referred to as 5th prime end of polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH. Sugar has a free OH of 3' group, which is referred to as 3' end of the polynucleotide chain. The backbone of the polynucleotide chain is formed due to sugar and phosphate. Backbone is due to sugar and phosphates. The nitrogenous base linked to sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group at second carbon, at second position of the ribose. Also in RNA, the uracil is formed at the place of thymine, RNA, and thymine is also called 5-methyluracil, another chemical name for thymine. Okay. DNA as an acidic substance present in nucleus was first identified by Frederick Miescher in 1869. He named it as nuclein. However, due to technical limitations in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on RNA diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing in the two strands of polynucleotide chain. However, the proposition was also based on observation of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine are constant and equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore is the sequence of bases in one strand is known when the sequence in other strand can be predicted. Also, if each strand from a DNA, let us call it as parental DNA, act as template for synthesis of a new strand. The two double-stranded DNA, let us call them as a daughter DNA, thus produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA become very clear. The salient features of the double helical structure of DNA are as follows. It is made up of two polynucleotide chains where the backbone is constituted by sugar and phosphate and the base projects inside. The two chains have anti-parallel polarity. It means if one chain has polarity 5' to 3', then the other has the polarity 3' to 5'. The bases in two strands are paired through hydrogen bond, H bonds, forming base pairs. Adenine forms two hydrogen bond with thymine from opposite strand and vice versa. Similarly, guanine bonded with cytosine with 3 hydrogen bonds. As a result, always a purine comes opposite to pyrimidine. This generates approximately uniform distance between the two strands of the helix. The two chains are coiled in right-handed fashion. The pitch of the helix is 3.4 nanometer, or 34 angstrom, and there are roughly 10 base pairs in each. Here you can see hydrogen bonds are there. Okay. Consequently, the distance between a base in a helix is approximately 0.35 nanometer or 3.4 angstrom. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bond, confers stability of the helical structure. So here you can see adenine A, T and G, C. Okay. Sugar phosphate backbone and bases are inside. Here you can see from DNA to we are making when you make DNA to DNA, then this is called replication, and when from DNA to mRNA, this is transcription, and from mRNA to protein, this is translation. This is the central dogma. Okay. Like I am making DNA from DNA, so this is replication. Okay, and when I would make mRNA, then this is called transcription, and when I would be making protein from this structure, then this is called as translation. Okay. In some viruses, what we see is flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to that process? Yes, this is called reverse transcription. And the enzyme is RNA dependent DNA polymerase. Okay, and the process is called 1 minute. Okay, reverse transcription. octamer core of histone molecule and a nucleosome is being formed. Okay, so DNA helix. Taken the distance between two consecutive base pairs as 0.3 nanometer, 0.3 into 10 to the power minus 9 meter is the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with the distance between two consecutive base pairs, that is 6.6 into 10 to the power 9 into 10 to the power minus 9 into 0.34. It comes out to be approximately 2.2 meters. Here I would show this to you. The number of base pairs with distance between the two consecutive base pairs. So if the base pairs is 6.6 into 10 to the power 9 and the distance between them is 0.34 into 10 to the power minus 9, then it comes out to be this one is cancelled and this one is also cancelled. So it comes out to be 2.2 meter approximately for humans. Okay. So a length that is far greater than dimensions of a typical nucleus, approximately 10 to the power minus 6. How is such a long polymer packaged in a cell? If the length of E. coli is given 1.36 mm, can you calculate the number of base pairs in E. coli? As we have previously seen that how many base pairs are there in E. coli. That here you can see E. coli contains 4.6 into 10 to the power 6 base pairs. Okay, so now we will come back to our question. So 1.36 into 4. Calculate the number of base pair 4. Okay, there is the length is 1.36 mm equal to 0.34 into 10 to the power minus 9 into x, and you can calculate it by using this. Okay, so x equal to 1.36 into 10 to the power minus 3 upon 0.34 into 10 to the power minus 9. So it comes out to be 4 into 10 to the power 6. Okay. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some proteins that have positive charges in a region termed as nucleoid. The DNA in nucleoid is organized in large loop held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires positively a charge depending upon the abundance of amino acid residues with charged side chains. Histones are rich in basic amino acids residue lysine and arginine. Both the amino acid residue carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules of histone octamers. 8 molecules of histone octamers. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as nucleosome. Typical nucleosome contains 200 bases of DNA helix. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. When nucleosome condenses, then what it forms is a chromatin thread like strained colored bodies, thread-like stained colored bodies seen in nucleus. The nucleosome in chromatin are seen as beads on a string structure which, when reviewed under electron microscope, theoretically, how many such beads nucleosomes do you imagine are present in a mammalian cell? The beads on a string structure in chromatin is packed to form chromatin-like fibers that are further coiled and condensed. Chromatin-like fibers are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as. The packaging of chromatin at higher levels require additional proteins set referred to as non-histone chromosomal proteins. In a typical nucleosome, some regions of chromatin are loosely stained and stains light and are referred to as euchromatin. The chromatin that are more densely packed and stains dark are called heterochromatin. Euchromatin is said to have transcriptionally active chromatin, whereas heterochromatin is inactive.